This presentation is going to be covering gene expression and regulation in eukaryotes. Gene regulation is the process that leads to gene expression and is effectively just the process of turning genes on and off, which allows cells to not express all of their genes in equal measure all of the time, which is logistically useful because some proteins are needed more than others or needed to function only in certain conditions. It also allows for different genes with the same genome to express different amounts of proteins and different types of proteins, which can be good for differentiation in eukaryotes. And even in prokaryotes, we see different immune evasion by producing different factors and changing up antigen expression in bacteria, for instance. In eukaryotes, there are multiple mechanisms for gene expression, for gene regulation. Epigenetics, transcriptional regulation, translational regulation, and post-translational regulation. Starting in with epigenetics, so to understand the basics, DNA wraps tightly around histones forming chromatin. The histone tail is rich in positively charged amino acids, which attracts the negatively charged DNA and keeps it bound really tightly. So there are two things we can do for epigenetic modification. We can modify the histone, in which case we modify the tail. There are two processes in this. There's acetylation, which adds acetyl groups to neutralize the positive charge of the histone tails, which in turn leads to a weaker interaction with the negative charge of the DNA and leads to it being less tightly bound around. There's also methylation, and this is the addition of methyl groups and can activate or repress transcription depending on location. We can also modify the DNA. Modifying the DNA is stronger than modifying the histone, so it's a more long-term modification. Methylation of the DNA adds methyl groups to cytosine bases on DNA, and as mentioned before, this is a longer-term modification than modifying the histones. And the less tight binding of DNA around the histones makes it more accessible for transcription later. Just like if you have a tightly bound knot, it's harder to get into it and modify the knot or untie the knot. All right, transcriptional regulation. This is regulation of the transcription process itself. We have a few key players here. There are promoters, and promoters are the binding sites for polymerase and transcription factors. So the polymerase is going to bind there and start the transcription process. There are silencers, which are the binding sites for repressors. So a repressor binds there, which prevents polymerase binding to the promoter, so the polymerase can't start that transcription process and give us the mRNA that we want. There are also enhancers. Enhancers are the binding sites for activator proteins. They encourage binding to the promoter, so polymerase is more likely to go to that promoter and start making the mRNA that we want it to. They can also be pretty far away from the promoter, like really far away on the linear sequence. And there are mediator complexes which connect the activator regions, the activators in the enhancer to the promoter region. So the process of an enhancer, because it can be pretty far away, the activator proteins bind to the enhancer. The DNA loops around, which allows the mediator complex to bridge the gap between the promoter and the enhancer, as seen in that image in the bottom right. Basically, the DNA loops around, though the enhancer and the promoter are now quite a bit closer to each other, and so they can be bridged by that mediator, which allows it to activate. And the proteins that are now in proximity to and or interacting with the promoter then boost polymerase activity, again leading to RNA polymerase to come in, attach to the promoter, and start making the mRNA that we want. All right, translational regulation. Both methods of this affect the mRNA in the cytoplasm. The first are repressor proteins. These bind to the 5' end of mRNA and prohibit their attachment to the ribosome. The second is microRNA or miRNA. These are small sequences, usually about 22 nucleotides. They bind to a complementary piece of messenger RNA, and if it's a full match, they just outright destroy the mRNA. If it's a partial match, they just prohibit its attachment to the ribosome. All right, next up, post-translational regulation. This affects proteins after they've already been synthesized. We can modify the shape and thus the function via chemicals. Some of these processes are glycylation and phosphorylation. So in phosphorylation, we add that phosphate group. It changes the shape and thus the function of the protein that we just phosphorylated. Another method that we can use to regulate this is protein degradation. Basically, the protein is tagged with ubiquitin, and that tagged protein goes to a proteasome, which unfolds and cleaves it into smaller parts, both small peptides and just amino acids. And this recycles it for later use, just like scrapping old stuff and using the parts later. Thank you.